anemia is just a decrease in the numbers of red blood cells in the body, and we focus primarily on Pcv and hematocrit. And we compare these to our reference range to decide whether or not a patient's anemic. Pcv is something that you can determine with a microhematocrit tube and a centrifuge, and I'm sure you've done that before if you've ever worked in practice. Hematocrit is something that's calculated from other numbers that are measured on a hematology analyzer for a complete blood count. Other things that are measured that tell you about red cell numbers on a hematology analyzer and CBC include the red cell count and then the hemoglobin concentration. And although we may glance at these, we don't really use them to tell us whether or not an animal is anemic. PCV and hematocrit tend to be a little bit more meaningful to us. So with anemia, the body then senses this decrease in oxygen, because there's, of course, if red cells are supposed to be carrying oxygen, there's less oxygen than being carried. And this is recognized by the kidney, which then secretes erythropoietin, which goes to the bone marrow, and then the bone marrow responds with increasing erythropoiesis and making more red blood cells, so that in 2 to 3 days in all species, including us, but not the horse, you will see an increase in early red blood cells in the peripheral blood. And so these are actually called reticulocytes that are released into the blood, and we're going to talk about those in a different lecture. So these reticulocytes are released into blood within 2 to 3 days, and they peak at around a week to 10 days in production in response. And so when you have an increase in reticulocytes, that's called a regenerative anemia. And when you have a lack of an increase, it's called a non-regenerative anemia. And so how we kind of classify anemia is based on this scheme, and it helps us determine the actual cause of the anemia. So regenerative types of anemia have to do with blood loss or red cell destruction, known as hemolysis. And non-regenerative anemias are due to decreased production, which is self-explanatory, I think. And so reticulocytes, except in the horse, are one of the things you're going to look at to actually tell you what's going on and why the anemia is occurring in an animal. So as we go on in the course, we're going to use several other criteria to help us characterize anemias. And I'm not going to go into detail in them other than to list them. One is the severity of the anemia. That can sometimes give us information. We're going to look at the protein, so what's happening in the plasma protein or the serum protein. We already talked about reticulocytes, so the regenerative status. We're going to actually look at who's our patient, right? So a young patient might be more likely to have GI parasites, whereas an older patient might be more likely to have neoplasia, which is obviously overgeneralization. We're going to look at actually what our red cells look like. So are there characteristic changes in our red cells that tell us about the etiology of it? We'll look at something called red cell indices, which are the MCV and MCHC. We'll look at bilirubin, so our actual patient's bilirubin, which can tell us, bilirubin being a breakdown product of heme, can tell us sometimes in some cases of injury and destruction of red cells. And then we'll look at things that tell us about kidney function to start with.